Welcome to Hartline Biology, reminding you that biology has its own language, and we love to name everything in biology. So learn the language, folks. Please subscribe to the channel. Let's start off with a recap of DNA replication. The goal of DNA replication is to create an identical copy of DNA. In order for this to happen, the DNA strands must separate. Then, new DNA is built to complement to the bases on the existing DNA strands. Where there's a G, a C nucleotide is added. Where there's an A T nucleotide is added. Notice the strand is growing from 5' to 3', and this happens on both strands so that now we have two identical copies of DNA. Well, what makes this happen? DNA doesn't do this all by itself. What's making this happen? Enzymes! Enzymes are little molecular machines, and we name them in ASE, or A's. Now, in order to go through these enzymes, we're going to simplify this diagram, and instead of showing all the nucleotides all throughout the DNA, we're going to show the DNA as two strands, like this. We're going to start off with DNA helicase, a super important enzyme which unzips DNA so that it goes from double-stranded to single-stranded. In order for this to happen, DNA helicase goes between the bases on the two strands and breaks the hydrogen bonds between them. Let's watch DNA helicase in action. It finds an area of the DNA known as the origin of replication, and then it unzips the DNA, separating double-stranded DNA into single-stranded DNA, forming what's known as a replication bubble. Topoisomerase is another enzyme that works together with DNA helicase. It helps preventing what is known as supercoiling. So DNA is not two strands that just run along side by side. They actually form a helix and twist around each other. Now, if DNA helicase was to unzip DNA all on its own, what you would get is something called supercoiling, where the helix will wind and wind and wind and create this supercoiling effect, preventing DNA helicase from continuing to unzip the double-stranded DNA. Instead, we have this enzyme known as topoisomerase, which uncoils the double-stranded helix so that DNA helicase can continue to unzip the DNA. DNA polymerase 3 is arguably one of the most important enzymes in DNA replication, because this is the one, folks, this is the enzyme that builds new DNA. But it requires what's known as a primer in order to get started. It can't start all on its own. So what is a primer? It's a short piece of nucleic acid, either DNA or RNA, and it's single-stranded. In DNA replication, RNA primers are used. Let's watch DNA polymerase 3 in action. So helicase unzips the DNA, a primer is formed, and there's DNA polymerase 3 building new DNA that complements to the existing strand of DNA. So how does that primer get there? We've got an enzyme for that too. It's known as primase. Primase is the enzyme that creates the RNA primers. Let's watch primase in action. Primase comes in and builds a small segment of RNA. DNA polymerase 1 is responsible for getting rid of those RNA primers. So the primers are just temporary. And then DNA polymerase 1 comes in, removes the RNA primer, and replaces them with DNA. There goes the primer. Let's watch that one more time. Primer's there, that's RNA, remember? DNA polymerase 1 comes in, removes the RNA primer, and replaces it with DNA. Let's watch this all together. Primase comes in and builds the primer. DNA polymerase 3 builds new DNA. And then DNA polymerase 1 removes the primer and replaces it with DNA. The last key enzyme we need to talk about is DNA ligase. Ligase connects gaps that occur between the new DNA strands. There's many reasons why you get gaps. More about that in the next video. Here's the new DNA. Notice that there's these gaps known as nicks between the new segments of DNA. DNA ligase comes in and fixes those by creating covalent bonds between the adjacent nucleotides to form continuous strands of DNA. Let's watch DNA ligase one more time in action, fixing all of those nicks. Thanks for watching. Don't forget to subscribe to the channel.